Okay, hello everybody. Thanks for coming to Metagenomics Lesson 1 for the BioVC Net. I'm going to talk about metagenomics in general as an introduction, but first I want to give an opportunity to any of the instructors on the line who would like to introduce themselves. I'll start. My name is Eric Collins. I'm an assistant professor at the University of Manitoba in Winnipeg, Canada, and I love bioinformatics. There are a number of other instructors who are working on putting together tutorials and lessons coming up later in the semester. So they'll be introduced later when they have those to share with you. So metagenomics is one of the funnest parts of bioinformatics for me, and I'll tell you a little bit about why that is. But the first thing I like to remember is just how complicated microbial communities are. And generally, I'm a microbial ecologist, and I think a lot of the people here are as well. But it's important to remember that in any community you go look at in nature, there's a whole host of things going on. In seawater, you have phytoplankton and bacteria and viruses and ciliates, protists, diatoms, all in all these different micro niches, whether it be on a piece of detritus or something else sinking through the water. You have the same thing going on in soils, where you have thousands and thousands of species coexisting, competing, working together. And we're learning a lot more about gut microbes as well. So the tract from your stomach to your anus contains many different environments, and different microbes are prevalent in different parts of that system. And so one of the things that we want to do using metagenomics is to, first of all, get an idea of what the organisms are there and what they're doing. Those are kind of the two main questions we ask and answer using metagenomics. So hopefully this will help you get started in asking and answering those questions. So the first thing we'll talk about is what do we mean by metagenomics? Then we'll go into what kinds of questions we can use metagenomics to answer. There's some that are really good uses of metagenomics, and there's some where other techniques might be more useful for you. So we'll talk about that in Is Metagenomics Right for Me? And we're not going to talk about the wet lab component, so we're not going to specifically discuss which kits you should use, what providers you should use to do your sequencing, stuff like that. We're going to assume that you have data in hand. But if that is an interest to a lot of learners, then it's possible we could have a roundtable or even add maybe a couple lessons on that. I'm not an expert on that, so here we're going to start with assuming you have data. And if you need help finding data online, we can also talk about that too. So we'll start that at the end. So what is metagenomics? We'll start by saying what we don't mean by metagenomics, which is 16S rRNA sequencing. In this network, we'll call that amplicon sequencing, and there's a whole channel dedicated to that. So Illumina has been, unfortunately, kind of egregious in spreading this term, 16S metagenomics, but here we won't be using that. We'll only be referring to metagenomics in its form as shotgun metagenomics. So what does that mean? Amplicon sequencing is the technique, also known as metabarcoding, where we amplify a small fragment of DNA from a mixture of many different taxa using a primer that's specific for that gene or target. So by amplifying a small fragment of the DNA, then we can specifically sequence that target and do downstream analyses like phylogenetic analyses or classification, things like that. This differs from metagenomics, which is shown in the bottom panel here, or shotgun metagenomics, which is where we add or take an environmental sample that already has many different taxa in it. We grind them all up and then basically sequence as much as we can afford of the raw data. So the most powerful instrument that people are using now to do that is called the Illumina NovaSeq, and it generates about 6 trillion base pairs of DNA per run, and you can get one of those for about 20 grand. And you can multiplex a whole bunch of samples, so you can say run 384 samples all at the same time and get a whole bunch of data. And so that's what we mean when we talk about metagenomics. In this case, we're talking about incomplete sequencing of a mixture of genomes using an untargeted approach. And why do I say incomplete? Well, that's because the environment is extremely complex and information dense. So just thinking about one single drop of seawater, a milliliter of seawater contains about 1 million bacteria. Each of those bacteria has a genome that's about, on average, 3 million base pairs. And in fact, a lot of them are dividing at any given time. So they may have 2 or 3 or even more copies of their genome in their cells that are being actively replicated at any time. In that same drop of seawater, you'll have something on the order of 10 to the 3 eukaryotes, so protists, diatoms, dinoflagellates, things like that, and we'll give them an average genome size of 300 million base pairs. So if you add those up, that's most of the DNA there is in the form of bacteria. We could also add viruses, but they add a relatively small amount because most of them are quite small. So on average, you might have something on the order of 3 tera base pairs per milliliter of seawater. Well, I just told you that the most powerful sequencing machine we're using right now generates about 6 tera base pairs of DNA sequence per run. So that means that you could sequence maybe 2 milliliters of seawater completely on a NovaSeq. So $10,000 per milliliter of seawater if you wanted to sequence it completely. As far as I know, no one's ever done that. Mostly what we do is we filter perhaps a liter of seawater onto a filter, extract the DNA, and then sequence a fraction of that, probably a very small fraction, maybe only a few percent. So it's important to realize that the output of a metagenomics sequencing run is not complete. It is only a partial representation of what's in the environment, and therefore the data you get will be compositional, meaning proportional, in that it doesn't represent a quantitative measure of what's in the environment. There may be ways to add standards, internal standards, where you can go on to do quantitative measurements, but that isn't the common thing being done right now. So we can go up to other environments too. So some of you may study soil. You know that there can be something like 10 to the 10 bacteria per gram of soil, a whole bunch of fungi, 10 to the 5 or 10 to the 6th. So in soil, there's something like 40 petabytes of DNA per gram. So that would be 6,000 NovaSeq runs, or about 2.5 to 3 times as much DNA as is currently deposited in the sequence read archive, which is about 40 petabytes of data right now. And if you look at stool, feces, it's even more. So there's something like 10 to the 11 bacteria, a whole bunch of eukaryotes in the form of fungi and protists, and human cells as well, the colonocytes. So there's something like 700 petabase pairs of DNA in every gram of stool. So this is just a reminder that there's a lot of information out there in the environment, and we're only looking at a small fraction of it. And that makes it difficult in metagenomics to look at things that are rare. It's often good to remember that most of the things we see when we're doing metagenomic sequencing are on the more abundant side of what is out there in the environment. So what kinds of questions can metagenomics be used to answer? As I said before, most of the questions fall into two major categories: who's there and what are they doing? On the who is there side, there's a number of techniques within metagenomics that we'll talk about as we go through this course. One of them is marker gene analysis, which can be used to look for taxonomic diversity, phylogenetic diversity. We'll also be looking at metagenomic assembly and binning. So we can do things like identify novel genomes and genome diversity, and using those, find out more about what's in the environment. For example, a lot of groups are using these now to detect entirely new phyla of bacteria and archaea that we had no idea existed 5 years ago. On the other side, on the what are they doing side, we can ask things like, are there novel genes that are present, and what is the diversity of those genes, using gene prediction? And functional annotation can be used to look at the diversity of protein families and other functional pathways that are encoded in these genomes that are in the environment. So there's an entire BioVCN network dedicated to functional annotation under #functional annotation, where they'll dive especially into the techniques for how we do a lot of the stuff on the right side of this graph. And this course will be focusing mostly on things that are on the left-hand side of the graph, and then link you into the tools for doing the right side. So what kinds of questions can metagenomics be used to answer? We talked about these. Some of the ones that I find really useful are questions like, which genomes encode this gene that I'm very interested in? For example, I'm interested in the NIFH gene, that does nitrogen fixation. What genomes in the environment encode this gene is a question that we can ask now that we can do, for example, metagenomic binning. How the heck many prokaryotic phyla are there in the world? Metagenomics is especially useful for organisms that we don't know how to culture and are maybe even uncultivatable at all outside of their natural environment. So the only way we have to access what those organisms are doing is to look at their genomes, find out what pathways they encode, and make predictions from there. On the community ecology and functional side, we have questions like, what antibiotic resistance gene does this community encode? So that can be very important for pathogen and other kinds of medical questions that people might be asking, or how many different carbon fixation pathways exist in hydrothermal vents. There's a lot of uses for metagenomics that involve looking for things that we aren't sure are there, or that may be different from what we already know about. So discovery of novel pathways, discovery of novel taxa is one of the things that metagenomics does best, in my opinion, because a lot of other current methods are based on only looking for things that we already know about. So metagenomics really shines when you don't know what you're looking for and you can go exploring. So is metagenomics right for your question? There's a number of other techniques that are discussed in the BioVCN network, and you may look to them for answers in case these sound like things that you're doing. So if you need to detect rare genes or species, metagenomics might not be right for you unless you're very rich. If you have a primer that amplifies a gene of interest, it is probably much cheaper to go out and just do the PCR and look at the results than to try and sequence deep enough to detect it. Metagenomics can be very difficult if you have a lot of eukaryotes in your sample, again, unless you're very rich, partially because assembly of eukaryotes from metagenomes is very difficult, And the more complex the community and the genomes, the harder it is to get useful information out of it. You end up wasting a lot of reads on parts of the genomes that aren't commonly useful for functional annotation. Also, if you are looking to answer questions with many, many samples in the order of thousands, it may be more useful for you to go the route of amplicon sequencing, because the tools for doing highly multiplexed metagenomics are not very well established right now, although there's a lot of people working on them. For example, people are working on robotic analyses to do very, very highly multiplexed metagenomes. And so in the next few years, that will become a lot more common. But right now, it's not that available. Quantitative PCR is another option related to amplicon sequencing. For example, if you only care about the presence or absence of something, or if especially you want to quantify how many copies of a gene or a species is present in your sample, that's probably a better way to go than using metagenomics. We kind of skipped talking about genomics in this, but there's a long history of sequencing complete genomes of things that you can isolate and grow in the lab. So if you can isolate your organism of interest, it's probably a great idea to do that and sequence the genome first before going out and looking for it in the environment. And again, if you're working with eukaryotes, it's also probably a good way to go. There's been a lot of progress in the world of single cell genomics recently by cell sorting and sequencing of whole genome amplification cells. So if your interest is in population genetics or novel taxa or horizontal gene transfer in pangenomes, then single cell genomics might be a better way to go than metagenomics. And I think Maria Pakiadaki is on this call, and she could tell you a lot more about that. But people have found that mapping reads from the environment to single cells is better than mapping to a metagenome. And Maria, correct me if I'm wrong about that. Finally, you might try meta or transcriptomics. Oh, she says she'd be happy to give a lecture if there's enough interest. I would be interested in that. Finally, you might want to try meta transcriptomics or transcriptomics if you want the levels of gene expression, or again, if you're working with eukaryotes. There have been some nice studies, for example, in the Terra Oceans data set, where looking at the differences in gene expression across ocean regions is much more interesting than looking at the differences in the metagenome content of the organisms, because their gene expression patterns tell you a lot more about what they're actually doing than what their potential is, which is all we get from metagenomics. So there's an entire transcriptomics channel in the BioVCN. So there should be some stuff coming out of that soon that you can also go take a look at if you're interested in that. Okay, so you've decided that metagenomics is the way to go. You've got your data in hand. Now what? Well, this is what we're going to talk about for the rest of the course. And a lot of people have spent a lot of time thinking about this. And so that means that there are a lot of published pipelines for analyzing your data. And some of the questions we'll address in this lecture series are how to choose what to do at each step of these pipelines. Generally, the pipelines involve some kind of quality control step. They may involve a step where you correct the reads, you trim adapters, you remove host contamination. Then there's a step usually where we do merging or assembly of the reads. Then we go on to binning and annotation. But at every step of that, there's numerous different tools to use, and there's numerous different ways to do it. So there's some published ones, and Some of them are quite nice. So using a published pipeline is very well and good. What we're going to do in this is try and go through each step step by step so that you can see how each step is done. And then using that knowledge, you can go out and pick the pipeline that already exists that works best for you, or that fits with your already existing workflow. Okay, so this is the end of the introductory lesson. I just wanted to let you know what the future lessons in this track are going to be. The next few lessons we'll talk about taxonomic classification using metagenomic reads. Then we'll talk about metagenomic assembly, and then binning of the assembled contigs. So if you want some more information about that, you can go to our GitHub repo here under topic metagenomics. And then I just wanted to remind you of the other BioVCN topics that may be of interest to you. Amplicons is great for amplicon sequencing, functional annotation, transcriptomics. Network science is especially involved in the community ecology aspect of metagenomics. And then population genetics and comparative genomics, we'll talk a lot more about the phylogenetics and phylogenomic aspects of what you can do with the result from the metagenomic data that you have.